Wairere runga, wairere raro, wairere i ngā taumahatanga kei wangiri a tātou katoa, kia wātea, kia mahea, tihei, mauri ora. Kia ora everybody, welcome back. Hopefully you had a quick bite to eat and you've all rejoined us, and we have new people joining us. And that you found time to mix and mingle in the discussion session at 12:30. But yeah, back into it. Before we get into it though, I will just go over our technical notes again before I introduce you to our chair for this afternoon's session. Should you experience any technical difficulties, please use the live support. During sessions, you can access live support on the right-hand side or from the foyer screen outside of sessions. If you do experience any viewing issues due to your internet bandwidth, please check on the audio video issues text located right below the video and choose one of the backup streams which are lower latency. The virtual platform allows opportunities for you to connect with other attendees. We encourage you to explore the meeting hub and the exhibition stands in the break times. You may have noticed that we have some prizes up for grab. Click the win points button to get involved. There are some awesome prizes to be won. Throughout the conference, please submit questions in the live Q&A icon and we will answer them during the panel discussions at the end of each session. So yes, please fire those questions through. No doubt if you're thinking it, someone else is thinking it. So please fire them through. Otherwise, you leave it to me and our chair to ask the questions, and you don't want me to ask the questions because sometimes they're a bit intense is what I got told. Someone said, you're asking intense questions. Anyway, with that, I'll introduce you to our chair for this afternoon's session, Bridget Farnworth. So Bridget has a PhD in behavioural ecology and conservation biology from the University of Waikato, Hamilton. Bridget's research background involves understanding the behaviour of shipwrecks and examining if predation cues can contribute to their management at ecologically vulnerable areas such as eco-sanctuaries. At home in New Zealand, Bridget has enjoyed working with a broad range of native species, including New Zealand dotterels, kiwi blue ducks, and carnivorous land snails, and Auckland's tree weta. I mean, if we get time at the end, I'm going to ask you a lot about that. So hopefully people fire questions through for the presenters. Otherwise, I'll be asking Bridget some questions. And her favourite animal is still the shipwreck, though. Tēnā koe, Bridget.
Thank you.
So before we get into the other stuff, our guest speaker for this afternoon session is a friend of mine, Jessi Morgan. Jessi is the Chief Executive of the Predator Free New Zealand Trust. Jessi has been with the Trust since its inception in 2013. The Trust's mission is to inspire, empower and connect people and community groups all over New Zealand in the efforts to suppress and ultimately eradicate pest predators such as rats, possums, mustelids and feral cats. Before working at the Trust, Jessi led the $1 Million Mouse fundraising campaign, a joint project between the Morgan Foundation and DOC, raising $1 million to eradicate mice from Antipodes Island. The project was confirmed as successful in early 2018, an endemic species on the island now thriving. She brings business and corporate experience from her previous life as a head of operations at TradeMe, a role she held for 10 years. Tēnā koe, Jessi.
Kia ora, Tame. Thank you. Kia ora koutou, ko Jessi Morgan, tōku ingoa. Thank you, Tamo, for that introduction. I didn't realise you were going to have to read that full bio, so apologies for that. I'll make notes of that for next time. So as Tamo said, I kind of come from a business background, but I've been at the Trust for about 8 years now, which is quite a long time that I'm becoming an old hand in this conservation space. But the Trust is really passionate about making conservation mainstream and making the predator-free movement accessible to everyone. We want to make it easy for people to get involved, and once people are inspired to be involved, we want it to be easy for them to stay involved. I'm sure you all know this, but 2050 is an ambitious goal to rid New Zealand of rats, mustelids and possums, and to ensure our precious native species can thrive. The vision's really inspired New Zealanders, and thousands of people across the country are getting involved in a range of ways, whether it's trapping on their private properties in their backyards or farms, helping on local reserves or on the public conservation land. People are really eager to get involved. I think the cool thing about the predator-free vision is that it brings a conservation concept together very clearly. It's very tangible that people can take action and see change quite quickly. So people can put a trap in their back garden, catch a rat, and feel like that they're contributing pretty closely. We kind of think of it a bit as a gateway drug, that they get involved in trapping in their back garden and they start to notice their local environment around them, their natural environment around them. So they suddenly start to realize there's more tui in their back garden, or the plant native plants to feed and provide habitat for native species. It also connects communities. So what we're seeing across the country actually is people starting in their backyards and it's spreading out from there and becoming a really concerted community effort. So we like to think of it a little bit like it's creating resilient communities, linking them more to their natural environment, and a little bit like Neighbourhood Watch used to be back in the day, for those of you who are old enough to remember that. And I guess the cool thing is, sorry, I forgot. I guess the cool thing about it is there's a role for everyone. So whether you're planting in your back garden, or you've got a bird feeder, or spending hours out in the back country checking traps, there really is a role for everyone in this, businesses included in that. So we recently undertook a survey of New Zealanders. So this is a representative sample, just over 1,000 New Zealanders. And we were really heartened by the results. So 75% of people strongly believe that New Zealand, Aotearoa, has native species worth protecting. And 57% believe introduced predators are a significant conservation problem. So we have captured that. People are starting to understand the issue that predators create, and that if we remove predators, then our native species will thrive. These questions, what's quite fascinating here is the high proportion of New Zealand Māori that are representative in these numbers, and they're much higher in their connection to te taio and to predator control. So that's very hard to see. However, one of the things we do need to think about is the context that we're looking at. And this data of what other societal concerns are in people's mind was quite challenging, I guess, because there's so many things that are vying for people's time and attention at the moment. And so in some ways, conservation takes a hit because of that. So we really need to think of ways to keep biodiversity loss and the protection of native species top of mind for people. People can really only keep kind of 5 or 6 issues in their mind. So how do we raise predator-free and the impacts of predators in that list of concerns? I was heartened to see that we're kind of on par with the COVID pandemic, and given how much publicity that's had recently, I thought we were actually doing pretty well. But there's also another study of this which shows you the top 10 concerns. Biodiversity data doesn't rate in those top 10 concerns, but you'll see on the right-hand side that the loss of biodiversity is now rating in this recent survey at 40%, people's concerns 40%, whereas in 2020 it was 29%. So it is climbing. People are becoming aware of it. Given this context, we really need to think about how we can keep people engaged in the mission, and everyone that we inspire to get involved. We really need to ensure that we keep them on board and support them in any effort that they're willing to put towards the mission. Unfortunately, in community conservation, it feels like only the strong survive. People need to be really determined and persistent to ensure their project is successful and that they can realise their community's conservation vision. And you hear a lot about the red tape and the barriers and the hurdles that groups are facing, and it really isn't a surprise when you hear those stories that only the strong survive. So one of the things we like to think about is how do we take a customer-centric view of conservation? It's probably a bit of a change to how we've worked in the past, but we need to start taking a customer-centric approach and really change our mindset to make sure we're serving the end user. Often volunteers, are really busy, they're donating their time for free. So what can we do to smooth that pathway for them? Any private company will tell you that to be successful, you need to focus on the customer. And I think in essence, what we need to do is we need to view ourselves as in service to community groups and people that are volunteering their time. So how can we best support community conservation? So this is a report that's come out recently from the Cawthron Institute, and this tells us some of the barriers that community conservation groups are facing. We also did a review, a similar report back in 2018 with Marie Brown. She was Brown then and now she's Dool, so Marie Dool. And when we did that report, she said to us, there's nothing really new in here. It's already been reported. And that's true. Like there are a number of reports prior to Marie's report in 2018 that show us the issues that groups are facing. The challenge we have is actually affecting change. We do multiple reports because they're relatively easy. You outsource them, get them done. They tell you what needs changing, but they don't result in any change. So how can we take these pieces of work and ensure that there are people that are willing and able to affect change in the sector to make some of these changes happen. I changed too early, but that's okay. So how can we take meaningful action, and how can we ensure that these reports are commissioned? Have people listening to them at the other end. One way, I guess, of doing this is to ensure that agencies and organisations that are supporting community conservation have a collaborative and connected approach. Imagine if all the funding reporting was aligned, due at the same time, and asking the same base set of questions for the same time period. That seems simple. It would save community groups a huge amount of time, yet we don't do it. We stagger our reporting times by a month here and a month there, so community groups can't reuse the same reporting. Imagine if we could do the same thing for funding applications. People could submit one funding application with maybe some targeted questions for the certain funds, but that the same application could be used multiple times. We just need to think smarter in this space. We know that groups that have paid coordinators attract more funding, keep volunteers longer, and are generally more successful. So we also need to think of the types of things we fund. We need to fund administrative and support roles to ensure these community groups are successful, and not just equipment and capital costs on the ground. To achieve predator-free goal, we're going to need to work together. No one agency or organisation can do this alone. So we need to share our intel. We need to break down the silos and ensure we get the best results. Agencies working in silos or competing with each other will do the whole movement a disservice. So as a friend of mine likes to say, there's no IP in predator-free. It's really hard to get people inspired. As we saw earlier, you can see the things people are grappling with, and as a movement, we're kind of competing for people's attention. So once we get people inspired, we need to make it really easy for them to stay involved. We can't afford to put up barriers and lose them. And so we just need to take that customer-centric approach. And one thing I think maybe we should all do is join the committee of a community conservation group, because it will really give us some intel in terms of what things are like on the ground. So anyone that's undertaken predator control on the ground knows how hard it is. You travel to these quite remote places. You spend the whole day carrying a heavy pack. You're clearing out quite grotty, manky predators from the traps. It's not for the faint-hearted. And part of keeping people involved and inspired in this movement is sharing with them kind of milestones that things are being done and that progress is being made towards the ultimate goal. We need to think about how we can do that. and how we can share information with people on the ground to give them some light at the end of the tunnel, that their job will be getting easier, and that when they stop doing this work, it's not just going to all be undone. So sharing some of the research that's being undertaken by scientists can really provide hope of the progress and the much needed light at the end of the tunnel. Remember that a lot of these groups have been using the same tools and techniques for 20 or more years, and they're hungry for change. They know that their tools aren't ideal. So what can we share with them that might improve their success rate? But scientific research isn't necessarily set up in a way to get the findings to the end user in a timely manner. Often there's a long lag between the actual research and the public article. So, and the other problem, I guess, with the published article is not many of these conservation volunteers won't be subscribed to those articles, so they might not even ever come across that research. So how can we better connect researchers with the people on the ground, and how can we connect the people on the ground better with the outcomes of that research? Groups are really hungry for this knowledge and want to know what research is being done. It keeps them really motivated, and it keeps them inspired to try different things with their predator control. When we profile science at the Trust, it's our highest rating topic. It was a real surprise for me. I thought people would like the people interest stories and human interest stories and stuff, but if there's a science article, that is what where all our traffic goes. So that they're hungry for it. So I'm really interested to hear how we can bring some of those early findings out and share them with community groups and the people working on the ground. What can we do to broadcast it to an even wider audience than we already have at the Trust, and how can we profile some of the science that you guys are doing? Research summaries, updates of of how research is going, and early learnings would all be really warmly welcomed. So I guess in summary, if we all worked together on sharing the knowledge that we're gaining and breaking down the silos, then surely we're going to get to this predator-free vision even faster, and the path will be smoother on the way. Hopefully, the takeaway for you is the catchphrase of "there's no IP in predator-free." Often in the space, people are really protective of the knowledge they have. Even at community group level, they're protective of what bait they're using in their trap or their secrets to success. So how can we start encouraging that sharing and making people feel like we're all in this together? Thank you.
Thank you, Jessi. So for those of you listening, if you do have any questions, feel free to flick them through. Oh, I see Desi's flicked one through. I'll ask Desi's one first because I've got a ton of questions here, Jessi, for you. And I know that, yeah, well, if we don't ask them here, I know I can easily email you them. So you might get an email from me. Kia ora, Jessi. Have you completed hackathons to connect communities to scientists to develop solutions?
No, we haven't done that. I think it's an awesome idea. I know in the tech space it's done to great success, and I think it would be a really good thing to do. So if anyone would like to help coordinate one, get in touch, because we'd love to support that happening.
Excuse my ignorance. What's a hackathon?
So you have a problem, and you get a bunch of people kind of from a range of different backgrounds, and they come together over a short period of time to solve a problem. So in a tech development space, it might be, solving a trap data capture thing, and people can build a system. So,
interesting. Never heard the word. The concept obviously is a wānanga, a workshop, but
yeah.
Cool.
Yeah, but it's kind of they're driving for outcomes that are very quickly, trying to they've got 24 hours and you've got to get to the end point.
All right, following on from that, you mentioned, you kind of finished up on it about science seems to be really something that people a lot of appetite for. What was the number one science article that you shared, or what was was there a standout?
That's a really good question. I don't know what our top one is. People are fascinated by animal behavior. They're fascinated by lures that are working. I know that in our webinars, the hedgehog webinar was really successful. We recently had one featuring Helen Blackie with all the work that she's doing in this space. Helen's amazing because she's really good at sharing what she's working on quite early in the process, and people love it. So that was another one that was really successful.
I agree with the statement about there's no IP in predator-free 2050. However, is there some guidance around, especially for something like mātauranga? I mean, you know more than anyone. I'm happy to share any mātauranga I have for it. Do you offer any guidance on how they can, how communities can adopt and implement mātauranga in an appropriate way?
Yeah, it's something we'd like to do more of, Tame. So I think communities are hungry for it, and we do encourage communities to kind of reach out to their local marae and talk to the kuia and kaumātua there to hear about their aspirations and understand what used to be in that area, hear their stories. But we don't really offer any guidance in particular other than just encouraging them to do so. But people are hungry for more. So if people have got any mātauranga or things they would like to share with a broader audience, we're definitely keen to hear from you.
Oh, I know a few people that would love to share their mātauranga, their whakaaro, so I'll start lining them up for you.
Awesome.
Some of those numbers, those were cool numbers, the statistics, the 70-odd% and the 57% that agree with pests are a problem. And you kind of answered it when you said, competing with things like COVID. How can we get that number up? How can we get more people to sort of know or realize that these predators are a problem?
Yeah, I think there's a few ways we need to. I think within the predator-free space, we're really talking within our own bubble. So we've got quite a significant bubble, but we're still talking within, the willing. So how can we pop that bubble and get to the mainstream a bit more? Probably through the Fight for the Wild was awesome in terms of getting to a mainstream audience. So how can we do more of that? What else does that look like? Taking a real comms-based approach in terms of getting it out to mainstream media and different channels. People aren't going to come and follow the Trust page if they've never heard of Predator Free. So we need to kind of work out different ways to take them. But community groups and community champions are really powerful. So people on the ground that are well connected and passionate and coming to knock at your door and say, do you want to get involved in this? I mean, obviously it's a high touch method, but that's probably your mates talking to you about it is probably the most convincing way to encourage you to join or to put you off if you've had a bad experience, right?
Yeah, totally agree about the community champions. We see it as well in the Māori space, Māori communities. Question here from Aaron. In the years to date, how successful have you been in getting people to share their IP or learning? Is prevalence of patch protecting declining? And if not, are certain sectors more reluctant to share than others? What works in overcoming this?
Yeah, in the community space, what we've been trying to do is we've been running a whole lot of face-to-face kind of public talks. And so we have been doing those with a guy called Cam Speedy, who's an ecologist. And he has this amazing way of saying, this isn't my knowledge. This is stuff this knowledge is not owned by any one of us, but this is just stuff I've heard as I've been traveling the country. And every local area is different. And so you need to adapt it to what works for you. But just encouraging people to talk, I think that face-to-face is super, super important. Scientists have actually been amazing in terms of their willingness to share and help us produce articles and profile the work they're doing. But I think we've kind of been stuck in this situation of only profiling research that's had publications. And so we need to, and that can be quite a long time. So we need to talk and share some of the research that's happening at the moment. So that the lag's just not so big. All sectors. There's a lot of patch protection in this space for a common goal. It's quite challenging. So I'm not sure the history of that, but I think it's changing over time.
And this isn't me trying to bring the challenge into it, but I do think the challenges have been good at this space. And the bio-heritage, I mean, I was fortunate enough to be in Tranche 1 and involved in Tranche 2. And I am seeing that the science sector changing in response to the challenges. So ngā mihi kia koutou, kia rātou. The last question before we hand over to Bridget is, how do we keep the customer? You mentioned it a few times, and you started off with a really good idea that, you know, this predator management gets people hooked. It's the gateway drug. How do we keep them hooked if we are getting rid of those predators, so there's no more sort of buzz of catching things, and that we might not get these birds back until 10, 20, 50 years down the track? Any other solutions?
Yeah, it's really challenging. I live in Wellington, and so we have Zealandia, which is very convenient for public outreach. So the birds do their own public outreach and fly into people's back gardens. It's much harder where those species have been absent for a long time and will take a while to come back. But I think there's other things to keep people engaged. So as the trapping numbers reduce, people can start planting. They can start getting involved in citizen science around invertebrate monitoring or bird counts, or there's other ways to engage them and tell that story. And, you know, we're a long way off, really. So we're going to need to keep people vigilant about, in terms of their monitoring of introduced predators in their area. So there's lots of ways to keep them engaged. But, yeah, we just need to make sure once people are involved, we make it easy for them to stay involved, because at the moment we just make it, we make them feel like an inconvenience to us, and we need to flip that on its head so that we're happy to be supporting them and kind of honoured to be there.
What about the recommendation of sharing recipes for some of our endangered species to keep them interested? So if that's the end goal.
Yeah, love it.
You don't have to answer that. But Jessi, thank you so much for your time, for your kōrero. It's always a pleasure. So ngā mihi ki a koe mō o mātou rangi whakawhira I tēnei rā. And
Thanks,
now we'll pass over to Bridget to introduce our next section and our next speakers.
Hi, good afternoon, everybody. If you tuned in late, I'm Bridget Farnworth, and I'm chairing this, which is about novel tools and strategies for invertebrates. So lots of our current pest control strategies have unintended and detrimental effects on the surrounding environment. We need new tools to effectively control pests while avoiding these unwanted side effects. The Novel Tools and Strategies Invertebrates team aims to develop a new landscape-scale invertebrate pest management tool by 2024. While working towards this goal, they are partnering with Māori organisations to understand how new pest control tools can enhance kaitiakitanga and promote tino rangatiratanga. And with that, I would like to introduce our first speaker, Sara. And Sara has a background working for the Greater Wellington Regional Council, but was recently seconded to Victoria University of Wellington to work on the National Science Challenge. Her expertise are in the intersection between mātauranga Māori and quantitative science, and that's the topic she's examining for her PhD thesis. Welcome, Sara.
Kia ora koutou. Kia ora e te whānau. So yes, I'm Sara, Dr. Sara Belcher, and I'm from Te Arawa. I was born, however, in Kirikiriroa, and so I have claimed or adopted Parongia as my maunga and Waikato River as my awa. So today I'm going to give you a very brief introduction into ESAT. Now, ESAT is the tool that I created that came out of my PhD. And ESAT is it's a way of actually linking the quantitative data that scientists get to Māori perspectives, such as whakapapa Māori no Wairua. And this tool has been applied to the work in the National Science Challenge, and it's being used to model the outcomes of genetically modification of wasps to make them infertile and therefore control them. So this is the first page that you encounter when you're using ESAT. This is the metadata page. So this page holds all the information relating to the project. So it contains the project goals and aspirations, and it contains information on the people involved with the project, who the iwi are, who the key stakeholders are, and that sort of thing. You'll notice that there's a map in the middle of it. So the map is actually a live link to Bing Maps, so you can actually put on there pins that show specific features, where the site is, where the monitoring infrastructure is, and that sort of detail. So eSAT contains 5 pages that hold the data. This is an example. This is one of the pages. And in this case, this page here holds information on wasp population numbers. So in this instance, there are the streams of data relate to the nests per hectare, colony traffic, wasp nests, and the biomass. And the 2 rows down the bottom are used to multiply some of those things to create the biomass per hectare calculations and that sort of thing. So you can actually do a little bit of data modeling or data manipulation through eSAT. On your right-hand side, you'll see the small charts. Those charts show the data for each one of those data streams as it's mapped. Now you'll see a blue line in each of those charts. The blue line is the target, the management target, and the little black lines on the charts reflect the modelling outcome. So we've applied linear exponential modelling to the data to reflect where it should head into the future. So eSAT is a management tool, but it also is a modelling tool. In this particular project, this page here is the first page, which holds the information on the wasp populations. Page 2 holds information on the outcomes of the genetic modification. Page 3 holds the information on ecological data, such as the outcomes for kaka and invertebrates. And the final page was relating to the social acceptance and social perceptions of the genetic modification. So, and there can be, and also had a stream of data that related to the economic inputs of the control operations. So this is a tool that can actually handle data and information, social, economic, ecological. It can hold all of that data and use it in the same model. So what it does is it indexes, puts all of this data into indexes, which can then be combined into the model. This is the outcome of the model. Down on your left-hand side, you'll see a list of each of the 5 pages and the contents of each of those 5 pages. Each of those 5 pages, I'll flip back a minute. I forgot to say one very important thing. You'll see here, this is the Māori perspective that's been applied to the data in this page. In this case, it's mana atua. And you can weight the different data streams according to its relevance to the mana atua as specified by the iwi. So you can put a different perspective for each of the 5 data pages and different weightings, and you can compare one value system, maybe from the iwi, to another value system as applied by council, for example, and you can compare the outcomes for the overall habitat health and restoration outcomes from the 2 different value perspectives that have been applied. This is where it all comes together. So down the side here, you'll see the weightings that were applied to the different data streams. And in here, this is the perspectives applied to each of the 5 data sets. And this is the outcome for the overall habitat health as prescribed when wairua has been applied to the overall project, and each one of those 5 workings pages or data sets has been weighted at 100 in this particular instance. So you can see that it's not looking that good in this with these data settings that have been applied. So this is a close-up of the outcomes. So the data settings here, if we apply the perspective of whakapapa and the weightings changed for each of the 5 data sets as weighted in this particular way, this is the outcomes. If we apply tangata, for example, and the weightings are set at this, then we get a much better or much different outlook. So I think that's probably about as much as I can discuss in such a brief time. So yeah, ngā mihi tēnā koutou katoa.
Thank you, Sara, for that really excellent talk. Now we're going to have a talk by Rose, and Rose is an entomologist with an interest in invasion biology. She is currently based at Victoria University, where she is researching next-generation pest control technology for honeybee parasites as part of her PhD thesis. Thank you, Rose.
And I will be talking about my research on the use of RNA interference as a next-generation pest control method for varroa mites. Honeybees are the most abundant crop pollinator worldwide, but they have been suffering from severe losses in recent years. Varroa destructor is a parasitic mite that feeds on honeybees and spreads disease and is considered the biggest threat to honeybees. The Asian honeybee is the natural host of Varroa, but this mite has transferred over to the commercially utilized European honeybee. And as the European honeybee didn't evolve with Varroa, it is ill-equipped to deal with the parasite. In fact, if beekeepers don't actively treat their hives, the colony will die within the year. So this graph here shows the increase in honeybee colony losses in New Zealand over the last 5 years. The proportion of losses are shown by the white bars, and the losses due to Varroa are shown in red. According to beekeepers, colony losses last year were the highest they've ever been, with an estimated loss of nearly 110,000 hives. And the number one cause was Varroa. The most effective Varroa control methods currently on the market are organic or synthetic miticides, which are applied directly to the hive. Like many pesticides, these treatments can affect non-target species, including humans and even the honeybees themselves. The bottom photo shows hundreds of dead bees at the entrance of a hive that had been treated for Varroa using formic acid. Unfortunately, this does happen. Chemical treatments can weaken or even kill bees, but if beekeepers don't treat for Varroa at all, the colony will collapse, so it is a risk they have to take. The other issue with using miticides is Varroa populations can develop resistance to these treatments, which is a possible explanation for the rise in losses in New Zealand. Traditionally, 2 Varroa treatments a year is sufficient to control these pests, but beekeepers are reporting that they are now treating 3 or 4 times a year, making Varroa management much more difficult and costly. Over the last decade, research has been conducted on the use of RNA interference technology using double-stranded RNA as a novel control method for Varroa. This approach silences genes that are crucial for mite survival. The dsRNA in our study targets Varroa during their reproductive phase of their life cycle. So here is a photo of a mother mite with her offspring on the bee pupa. The bottom photo there of mites shows a typical mite family that would emerge with a bee when it hatches. We tested the effect of our dsRNA on mite mortality and reproduction. Though we didn't find an effect on mite mortality, we did observe a significant reduction in the number of offspring produced. Results are presented in this graph. The average number of offspring per mother mite is on the y-axis, and the different treatment groups are on the x-axis. So for our experiment, we tested a low dose and high dose dsRNA, shown in yellow and orange. As you can see, both doses were highly effective in reducing the number of offspring. The green and blue bars are for the 2 control groups. One was a non-specific dsRNA control, and the other is a plain sugar water control. As you can imagine, we were very excited about these results, as the dsRNA is controlling mite populations in a non-lethal way, and it specifically targets mite proteins, so it's harmless to honeybees, humans, and other non-target species. It's also non-toxic, it degrades in water and soil, so it doesn't negatively impact the environment, and it's approved by the EPA. So using Varroa as a model organism, we believe RNA interference technology has real potential as a control method for pest species. I'd just like to thank Greenlight Biosciences for their collaboration on the project and the National Science Challenge Committee. And thank you to Pike, who provided the data for our colony loss survey. And thank you to my supervisors, Phil and John, for their support in the process.
Thank you, Rose. And now we're going to hear from Symon Palmer. So Symon is based at Te Kawa-a-Maui School of Māori Studies at Te Herenga Waka, Victoria University of Wellington, where he is working as a researcher on the Bioheritage Challenge. Please join me in welcoming Symon Palmer.
Tēnā tātou, kei te mihi au ki koe Bridget and Tame. Thank you for your kind hosting today, and also a quick mihi to Sara and Rose for their presentations. So yeah, today I'll be expanding on the research that's already been presented, with an emphasis on seeking Māori perspectives. So just to contextualize our team, you will have noticed there's some quite different kind of topics going on so far. We have a work stream led by Phil Lester looking at Varroa and RNAi, which Rose presented from. We also have WASP and Gene Drive program with Peter Dearden down at University of Otago, and me and Sara represent part of the Māori Perspectives team. And you'll notice some asterisks next to all of these names, or most of them, and they are all early career team members, which is pretty amazing. So our program's really interested in fresh talent and actively building capacity. Just to reiterate some of our pests of interest, Varroa, which you've just heard about, and RNAi as a potential tool for control, and also the German and common wasp, which Peter Dearden and his team are interested in with Gene Drive. Now, both of these pests are invasive species. They aren't native from here, and they provide an intriguing point to ask questions on social acceptability for these new pest control tools. So with concern of seeking Māori perspectives, the topic, intersects and revives old issues, historical opposition to genetic modification, for example, of which there is a reasonable amount of literature on around the Royal Commission era, but also things like Y262, in which Indigenous Māori interests in genetic resources, the concerns for taonga and rongoa resources are vocalised. So this is kind of a part of the backdrop to our research program that we have to be aware of and navigate, and adding to that is the ill-equipped policy backdrop, which Arawa kind of spoke to yesterday, but specific to our work is the Has No Act, and also global interests in genetic tools across all sorts of fields, but also Indigenous concerns and interests in things like UNDRIP. So where does that leave us? Well, we're in the 2020s all of a sudden, and so we're wondering what the kind of current opinions are from Māori on these issues. So what is the pulse on these issues? It's kind of where we begin our socio-cultural research, and there's many access points to these complex topics, including social license to operate, theory, Te Tiriti o Waitangi, iwi hapu mana whenua, but also acknowledging the non-homogeneous identity of Māori in the contemporary era, tikanga Māori and historic resources and precedents, which we can find through literature reviews, etc. We also recognise a strong need for qualitative research in our program and the bridging of Māori views, mātauranga and Western science. So just going to quickly cover some of the work that we've been doing. We've had 3 distinct qualitative studies involving different sort of cohorts and organisations within that self-identify as Māori, including Māori businesses and an informed audience, Māori university students who had studied similar topics around biotechnologies and genetics, and also self-identified religious or spiritually affiliated Māori. So quite a diverse group of Māori interviewed using a kaupapa Māori influenced methodology, mixture of individual and focus groups, and also incorporating some mixed method tools like Likert and Q methods. So some of the themes that came out of qualitative research, unsurprisingly, there was a range of views, and I really do want to emphasize range of views. There were, for example, some views that permitted the use of these technologies were all on board, and then there was also strong opposition, which we're familiar with in some of the literature. But also some of the more interesting themes, do nothing. Do nothing is not an option. So people recognize that there are these biosecurity risks and current invasive species that are detrimental to biodiversity, etc. There's also concern for these genetic tools and off-target effects, and a desire for more information from a neutral standpoint, which is quite a difficult thing to think about sometimes. There was also references, unsurprisingly, to tikanga Māori, concerns for whakapapa Māori, kaitiakitanga, linking back to previous concerns as well. And consideration for humane approaches and invasiveness, sort of the scale of invasiveness of the pest, but also how some of these technologies may not actually hurt, like some of the trapping tools do. Taking some of our qualitative research, we recently did a second analysis of our studies and questioned, we've been questioning kind of the role of social license to operate in this kaupapa. So we were looking for a Māori framework to work from instead, and Hirini Moko meets take utu ia as a kind of starting point that we worked from. Take means to have a causal problem, and utu is an exchange of reciprocity or cost to reach ia, which is the kind of endpoint satisfaction and reduction of tapu from a problem. So this is kind of a Māori framework of thinking that we applied to qualitative data, and what that surfaced was the importance of a tikanga standpoint on this conversation, this very complex conversation, and also signaled a shift from social license to operate to rangatiratanga, which is probably more on board, as we are probably aware, with Māori interests in this space, conservation. And rangatiratanga provides this alternative lens on the issues where Māori issues and concerns more easily surface, and there's a little bit more autonomy over the conversation. Just quickly, another project that we have been working on is whakatoki as a source of mātauranga. So there were some cool presentations yesterday talking about mātauranga being in different mediums, and waiata karakia being some of those, but also whakatoki are another source. And so we were interested in historical precedents for a conversation of reaching predator-free 2050. And we wanted to know if there were traditionally pests for Māori, if there were pest management tools that were adopted, traditional tools, and if there was any evidence of eradication. And quickly through this review of whakatoki, I can say aye, and kao. So there were pests, particularly for kumara, which is a staple carbohydrate for Māori. And there was also pest management tools incorporated to control caterpillars and moths, for example, through fires and smoking, kawa, etc. But there's no clear evidence of eradication. And this is quite interesting when we start to think of the concept of a predator-free 2050 and these new novel tools, which kind of opens up further questions that we'd like to explore with participants. So leading forward, the pathway ahead, currently we're running a Māori beekeeper survey. So if you know a Māori beekeeper, please send me an email because we're trying to reach as many as possible. Currently there's no data on Māori beekeepers per se, but we're really interested in how what their pest management needs are and what their level of concern are for current pests. We're also hoping to work with some Māori beekeepers from this survey, identifying people that might be interested in partnering to work closer on these novel tools and learn more about them and raise any concerns. We also acknowledge from our research that there is a bit of a gap in communicating these complex scientific tools. So we're exploring some new communication tools and potentially looking at partnering with other work streams on delivering these. One of the ambitious and crazy things that we've been talking about is VR, which one of our research assistants, Te Amohaere, has had experience in explaining coded dieback to children, illustrating that virtually. So we see potential for these sort of technologies to help communicate tools, and also there are legislative considerations, as mentioned. So I think that's my 10 minutes. If you have any interests in our research, I'd love to hear from you. Drop me a line. Kia ora.
Thanks everyone for those really excellent talks this afternoon. That's been great. So just if you've missed it, we've had Sara Belcher and Rose McGruddy and Symon Palmer who have been speaking to us, and we're now going to have a panel session about each of their talks. So if you do have any questions, please feel free to drop them into the question box and I will try and make my way through them. Otherwise, we could just make a start, and I might just start with Rose. I had a couple of questions that popped up. Really interesting talk about Varroa. Roughly how much do you think that we would be spending on control of the Varroa mite at the moment, like dollar figures wise, ballpark? Lots is an acceptable answer too.
I think right. Well, I think it's $30 or $40 a year annually usually, and I think beekeep per hive, which is what it used to cost. But I think with the increase in applications and as well as the actual product itself, it's also paying people to go out into the hives and put the product in and then remove the product after a certain amount of weeks, because if you do leave a treatment in, it builds resistance. So paying someone to go put it in, bringing it out 6 weeks later and having to repeat that through the season, it is building. 150,000 a year for one beekeeper is the number I have just been passed.
Wow.
Yeah.
That's quite significant too. And so the potential for sort of your research in RNA interference really actually is quite significant, isn't it?
Yeah, and it would be a treatment where you just popped a pouch into a hive and the bees would finish it off and you wouldn't have to worry about collecting it until later in the season. Resistance wouldn't be as much of an issue.
Yeah, and so what do you sort of think it's going to take for that to lift off?
We need to do some more trials. We'd like to do some field trials in New Zealand. At the moment, it's just in the lab. Our collaborators in America have started with field trials though. So a little bit more to be done. We also want to get the opinions on beekeepers and how they feel about this kind of treatment, but hopefully just a few years and this might be something being used.
But we have proof of concept. That's very exciting.
Yeah.
We've got a question in the chat from Claire. We do have, I think my screen has locked, so it's That's not popping. I'm not sure. Tommy, are you able to see that in there? Because my screen has locked. Just perhaps while he's sorting that out, I might just perhaps invite Symon. Symon, you are co-leading Ngāi Karere, an early career network, and I just wanted to give you the opportunity perhaps to use this platform to tell anybody who doesn't know about it about it.
Yes, absolutely. Thanks for that. So if you missed yesterday, we had a quick presentation and a networking lunch where we kind of did breakout rooms and start to mingle as best as you can on these virtual platforms. But basically, Ngāi Karere is there to support any early career member in bioheritage. We're interested in hearing about diverse career paths, supporting people into those, and also providing professional development opportunities. So feel free to email me, once again, same email, simon.palmer@vuw.ac.nz, and if you want to get involved, if you missed any spam about yesterday's networking, then I can add you to the list.
That sounds great. Just while we're on that, here's an additional question for you, Symon. And apparently it is more of a comment than a question, but could district dock officers or the community rangers within them have contact details for Māori beekeepers, or at least those who have consent for hives on conservation land? Do you think that's perhaps an option?
That is potentially an option that I haven't explored. I have been spamming the TPK regional hubs to see if they know some people, but I'll definitely take note of that. Thank you.
Very good. And just while we're here chatting with you, we might as well have an additional question. I was really interested by your comments to do with whakatauki and what Māori considered pests originally. So were they just invertebrate? Were they the results of what you were looking at? Were they just invertebrate pests that were really highlighted?
Yes, yes, and that was a focus of our. We were looking for invertebrates in particular. I don't actually know about mammals.
I could help there.
Oh, there we go. Tama's in a similar field.
Lovely.
Well, I actually take over from that. I leave the creepy crawlies to Symon, but for vertebrates, yes, at one stage they did. So kirerea, what we often use as the Māori word for pest, is actually a variation of, kirehe, the word we kind of use for pests, is actually a variation of kararehe, which just means vertebrate. And so the first pests in that sense were the kiore and the kuri. In their own ways and rhymes and reasons at different parts of the country at different times.
And on that, this is probably a bit of an outrageous question, but do you think we ever had a stage where we had so many birds, obviously, perhaps prior to mammalian pests establishing themselves, do you think we ever sort of had a stage where we had so many birds that even they were sort of considered too loud and noisy? Did that pop up in any whakataukis? Probably not if you were just focusing on invertebrates.
I think, interestingly, the negative kind of traits of some pests were things like cicadas being noisy, for example. So that's kind of one example. But birds, more positive, like, there's whakatauki that talk about the embellished kind of vocals of birds and that being a compliment. So, yeah.
So no one's being annoyed by noisy bird song. Very good. And I might just pop over to Sara. The site that you had up, is that accessible to everyone? That's a live sort of site that's at the moment? I'm not sure if you might be on mute, Sara. You might just have to take that off.
Does it look like I'm on mute?
Oh, there we go. Now we can hear you.
Okay. No, it's a prototype at the moment, but one of my research goals very shortly is to make it a live portal. If anybody wants a copy of it, they're more than welcome to email me and I'll send them a copy of it. At the moment, we need to, me and my research assistant that helped build it, he actually built it according to my specifications, but both of us need to sit down and put a policy, well, an instruction manual together that I can go over it with people until that's done anyway. So it's very early stages of development at the moment.
Oh, very good. And actually on that, we have an additional question for you. We're interested to know how whakapapa was made into a metric that can be used in the data set. What was the process like of recontextualizing to our Māori concepts and ways of being into a metric for your framework?
Okay, well, that how I did it was the way that I view whakapapa. If somebody else was asked, they view whakapapa differently. So their description of whakapapa will be determined by their iwi or personal framework and background, and they can figure out, everybody who uses this can interpret these perspectives in their own way and then weight the data according to how they perceive those perspectives. So pretty much I asked people, okay, well, if you're looking at that tree and you're wanting to measure whakapapa and you're looking at that tree or that patch of forest, what exactly would you be looking at when you're thinking about whakapapa? And those are the things that we can then turn into something measurable. Like it could be the whakapapa of the invertebrates on the tree, it could be the whakapapa of the trees, the actual trees. So then you could actually determine how many of them are indigenous as opposed to introduced species, and that could be your whakapapa. So it depends on your interpretation, and there's no set way to determine it. You determine it according to what you want to, and ESAT can accommodate that.
Very good. We have another question about the Varroa mite. Is there genetic variability that can fight off the mite?
Not know of at this stage, no.
Very good. So everything is susceptible. Right, sorry, just trying to keep up with the questions here. Another question for Rose. How general can dsRNA be applied? Are there certain species or taxa you think would respond better to this method? And then the following, which we've already addressed, is how long does it take to go from research on dsRNA to using it as a control tool? So quite a bit there. Did you catch all that?
Yes. I think it has more success in invertebrates and plant pathogens and fungi. Vertebrates, it hasn't been found to be particularly effective this far for many species, but it is actually being used over in America actively to control potato beetles over there. And I think there are, in New Zealand, research is being done into dsRNA for controlling myrtle rust, and I believe that's going quite well at the moment. I know that this research, probably looking at Varroa control using dsRNA, began maybe 10 years ago, and there have been a few studies since then, but maybe the last 5 years actively using this particular method, developing it. So maybe, I don't know, 7 to 10 years is probably the time it would take from first trials to actually being a product on the market.
From woe to go.
Yeah.
Very good. Okay, we probably just have time for one last question for Symon. Do you see any links between rongoa Māori and pest management during your search for mātauranga in regards to pest existence and or eradication?
In short, no, regarding this project, and considering we're interested in the invasive species and maybe if there's a comparable pest in the whakatau that we just had a quick think, and probably not really. So the main pests and pest controls that were coming up were kumara plantation related, and they involved very localized control through fires, ahitapu, and also kawakawa and that sort of derived stuff that could be applied locally. So eradication, not really on the radar as and such. Yeah.
Fabulous. And I think on that note, we might hand back over to you, Tame.
Cool. Time's fast approaching for us to end. So big mihi to our kai kōrero. I don't think I'm going to get through my wrap up in 10 seconds, so I'll just keep it short and say thank you so much for sharing all of your whakaaro. I wish we had longer because we had a lot more questions, and yeah, we'll not get through all of them all. So, tēnei ka mihi kia koutou. To those of you listening, kia koe e tawhitito haere noho tonu mai. Stay with us, come back in 10 minutes. So go grab yourself a cup of tea, a coffee, and we'll get into the next session. Nei rā ka mihi kia koutou.